Welcome to our journey into the nervous system. Today, we are going to tackle a topic that everyone has experienced, but very few truly understand: the molecular mechanism of pain. Imagine your house has a state-of-the-art security system installed to protect you from danger. In your body, pain works exactly like this security system, and it is absolutely essential for your survival. Today, we are going to zoom in incredibly close to see exactly how these microscopic sensors turn a physical danger into an electrical warning signal. Now that we have introduced the idea of pain as a security system, let us understand why this biological alarm is so crucial. When you accidentally touch a burning hot stove, your skin needs an immediate way to detect that dangerous heat and scream at your brain to pull your hand away. In neuroscience, this process of detecting tissue damage or potentially damaging stimuli is formally known as nociception. Without this rapid detection system, we would not be able to react to environmental hazards, making it a fundamental mechanism for our self-preservation and survival. To understand how nociception works, we must look at the specialized sensors involved. The sensory neurons responsible for this crucial task are called nociceptors. They feature unmyelinated or thinly myelinated free nerve endings spread throughout your body. At the molecular level, the mechanism begins with a process called transduction. Transduction is simply the conversion of a noxious chemical or thermal stimulus into an electrical signal. This remarkable conversion happens because the membranes of these nerve endings contain specialized receptor proteins, most notably the transient receptor potential, or TRP, channels. While we often think of pain as a terrible nuisance, this brings us to why it is actually one of our most critical evolutionary adaptations. Without the molecular mechanisms of pain, we would constantly injure ourselves without even realizing it. There's actually a rare genetic condition called congenital insensitivity to pain, where individuals are born with mutated, non-functioning nociceptors. People with this condition often suffer from severe injuries and shortened lifespans because they lack this biological warning system. Furthermore, understanding these exact molecular gates allows doctors to design better painkillers and anesthetics to bring relief to suffering patients. Now that we know what these receptors are, let us look at how their opening actually creates an electrical signal. When a pain receptor, like TRPV1, is exposed to extreme heat or acid, it physically changes shape and opens up. This creates a temporary tunnel that allows sodium and calcium ions to cross the otherwise impermeable cell membrane. Because these ions carry a positive charge, their movement into the neuron creates a measurable electrical current. This inward rush of ions is the very first step in generating the electrical impulse that will eventually reach your brain. This flow of charged particles we just discussed can be described mathematically by the fundamental ionic current equation. This formula is essentially Ohm's law, adapted for the biology of a neuron. I-ion represents the actual flow of charged particles into the pain neuron. The symbol G-ion stands for the conductance, which tells us how many pain receptor channels are open. Vm is the current membrane potential, or the voltage inside the neuron. Finally, E-ion is the equilibrium potential, where chemical and electrical forces are balanced. If the painful stimulus is stronger, more channels open, increasing conductance and creating a massive inward current. Before we move on, let us see this formula in action with a concrete example. Imagine you touch a hot cup of coffee, opening TRPV1 receptors. We're given a resting membrane potential of negative 65 millivolts, a sodium equilibrium potential of positive 60 millivolts, and a conductance of 2 nanosiemens. We want to find the initial sodium current. First, we calculate the driving force by subtracting the equilibrium potential from the membrane potential, giving us negative 125 millivolts. Multiplying this strong inward pull by the conductance gives us a final answer of negative 250 picoamperes. In neurophysiology, this negative sign means positive ions are rapidly flowing directly into the cell. So, we have calculated the inward current, but what does this current actually do to the neuron? This massive influx of positive charge from the sodium ions rapidly depolarizes the neuron's membrane. Depolarization means the inside of the cell becomes less negative, pushing it toward a critical threshold for firing. Once that specific threshold is crossed, the neuron fires an action potential. This action potential is the actual electrical signal that travels all the way up your arm to tell your brain that you are touching something dangerously hot. The stronger the initial current, the faster this threshold is reached. This brings us to an important question. How does the brain know if a surface is just warm or dangerously burning? The intensity of pain you feel depends on how fast the nociceptor fires action potentials. This is described by a simple linear model, where frequency equals a constant K times the difference between the generator potential and the threshold voltage. The frequency F is the number of electrical spikes per second. The term in the parentheses represents how far above the minimum trigger point our pain signal is. If the generator potential increases due to a severe burn, the frequency goes up, and the brain interprets this as a sharper, more urgent pain. Now that we have this linear model, let us calculate the exact firing rate for a severe burn. We are given a threshold voltage of 15 millivolts, a constant of 4 Hertz per millivolt, and a strong generator potential of 35 millivolts. We want to find the firing frequency. First, we subtract the threshold from the generator potential, which gives us an effective stimulus of 20 millivolts. This means the stimulus is 20 millivolts stronger than the absolute minimum required to fire. Multiplying this by our constant of 4 gives a final frequency of 80 Hertz. This means the neuron is firing 80 distinct action potentials every single second, prompting your reflexes to immediately pull your hand away. Let us bring all these concepts together by looking at this visual summary of the transduction mechanism. Here, you are looking at a cross-section of the nociceptor's cell membrane. Notice the TRPV1 channel protein embedded right in the middle of the lipid bilayer. On the top, in the extracellular space, you can see triggers like dangerous heat, capsaicin, and acidic protons. When these stimuli interact with the channel, it forces the gate open. Look at the arrows pointing downward. They represent the sodium and calcium ions rushing into the intracellular space. This exact physical movement of positive charge is what creates the depolarization, or generator potential, that we just calculated, ultimately firing the warning signal to your brain. We have covered a lot of ground today, so let us review the key takeaways. First, we learned that nociceptors are specialized neurons that act as a vital biological alarm system, protecting us from severe injury. Second, we saw how TRP channels perform transduction, physically opening to convert dangerous heat or chemicals into an inward flow of positive ions. Third, we use the ionic current equation to calculate exactly how strong that electrical current will be. Finally, we discovered that the brain interprets the intensity of pain based on the frequency of action potentials. You now understand the complete molecular gateway of how we perceive pain.